Welcome back to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology Module 25. This is the beginning of our virology section, and it's our Introduction to Virology module. Welcome back to Falcon Physician Review's online review for USMLE Step 1. We're in Microbiology Module 25, where we're going to talk about viral structure. Viruses are obligate intracellular molecular parasites. Their genome is composed of either DNA or RNA. The virus genome directs the synthesis of virion components within an appropriate host cell. The virus can either have an envelope or be a naked capsid structure. Assembly of newly made viral components produces progeny virus particles. Progeny particles spread the infection to new cells. Viral structure is composed of a set of genes, which can be RNA or DNA, and a protective protein coat. Those two things together are called a nucleocapsid. Some viruses also have envelopes, which are lipid membranes they acquired from the host cytoplasmic membrane during their budding. If a virus is called naked, it means that it doesn't have an envelope and simply has the nucleocapsid. Viruses do not have organelles or ribosomes. They depend on us for that. Viruses are small. In fact, they're 100 to 1,000-fold smaller than the cells they infect. They have a protein shell, which is called the capsid, which surrounds the nucleic acid genome. They also have a capsomer, which is repeated subunits of capsids, which determines the shape of the virus. As I said, some viruses have envelopes, which is a membrane surrounding the nucleocapsid. Spikes are proteins or glycoprotein structures that jut from the virus surface and are highly antigenic. Capsid structures are made of repeating units of viral protein, which is the capsomer, and viruses come in many different shapes and sizes. You can have icosahedral structures, cubic structures, helical structures, or complex structures. The shape of the virus helps identify which type of virus it is. The icosahedral structure has a unique structure where all faces of the icosahedron are identical. It has tremendous symmetry. It's based on 12 corners, each occupied by a capsomer, and 20 regular triangular faces, which are hexons. Here's a picture of the adenovirus showing the icosahedral structure. You can see it does have some glycoprotein sticking out from the capsid structure. A helical structure is where protein subunits or capsomers interact with each other and with the nucleic acid, usually RNA. The length of the helix is determined by the length of the viral nucleic acid. This is the structure within negative enveloped RNA viruses. So helical is always RNA. Complex structure viruses have regular structures, but the nature of the symmetry is not fully understood. As you can see here, the pox virus is a good example of that. It's got all kinds of protein subunits all over the place. So this figure nicely illustrates the virus size and shape of various different virus families. You can see here they're classified according to their nucleic acid, whether it's RNA or DNA, double or single-stranded, positive or negative strand. They're also organized by complexity, by their capsid, by their envelope, and by their size. There are some natural consequences of the virus structure. If it is a capsid, it is resistant to acid solvents, it's resistant to drying, it's stable in the gut, and you can get a lot of fecal-oral transmission. It's released by cell lysis, and the antibody is protective against it. If you have an envelope virus, it's usually labile to acids and solvents. It has to stay wet. It can't go through the gut except for coronavirus and hepadnavirus. It's released by budding through the host cytoplasmic membrane, and cell-mediated immunity is required. When we talk about genome structure, it can be RNA or DNA, single or double-stranded, linear or circular, segmented or non-segmented. Examples of that are influenza has 8 segments and rotavirus has 10 segments. Wrapping up Module 25, this was just an introduction to virology. We talked about what a virus is, how they come in different sizes and shapes, and whether or not they have envelopes or whether they don't. Remember, the presence of an envelope will limit the ability of the virus to live in the outside environment. It also limits which tissues it can target. We're going to go on in the different modules and talk about specific viruses, the diseases they cause, and how we treat them. In our next section, 26, we're going to talk about viral replication.